All right, so here's video 2 for our benchmark review for the second 9 weeks. So what we're going to be looking at now is just as many as I can get in to the 9-minute video, and then I'll finish up in another video if I have to. All right, so it says number 14, it says, what's the missing from the box? Okay, so we're looking here and it's talking about photosynthesis and it's talking about cellular respiration. And if you look at most of these things, it gives you a clue to what's missing. Okay, so I know that photosynthesis and cellular respiration, both of them use glucose. That's C6H12O6. Both of them use oxygen. However, the problem is they both don't use ATP. So what's missing from the box is both glucose and oxygen. And all I had to do was simply look and see that that is what was missing. All right, the following is an example of what phase of mitosis. So when you're doing these, it's important that you look and you say, okay, mitosis means middle, into the middle of the cell. Okay, and so I can automatically look at this and I see that all my chromosomes are in the middle of the cell. That is indeed metaphase. Okay. Number 16, what's the best explanation for what's going on in the cell? So you've got two choices. It's a plant cell forming a cleavage furrow or an animal cell forming cleavage furrow. Now, a lot of people will like, well, how do I know the difference? Well, it's very easy because a plant cell needs to be square. Because in order for a plant cell, for a tree or a flower or whatever to stand up to wind and pressure, it has to be square so that they can stack on top of each other and so that they can be firmly on top of one another. So that's a plant cell, it would be square. The animal cells are circular because we don't have to withstand wind, or we don't have stems that have to stand up. So the only way that you could get this wrong is to not understand that plant cells are square, animal cells are circular. So in this case, the answer is B. Okay. A human skin cell has 46 chromosomes. After mitosis, each of these cells produced will have how many cells? Okay, now the key words here are mitosis. Okay, and in mitosis, we're talking about skin cells and liver cells and anything except for sperm and egg. Okay, so that means that whatever I start with, I end up with exactly the same because I have a duplicated cell. You don't want your skin cells to start changing and mutating. That's it becomes a problem. So in this case, the mitosis is the big keyword, and okay, for this cell to have being a human skin cell that it tells us, and that mitosis, and that it has to have exactly the same amount of chromosomes in the end than it does in the beginning, you have to be very careful with how you read this question because A says it's 23 pairs, then B says 46 pairs, and automatically you want to say, oh, it has to be 46 pairs, when indeed it's going to be two cells with 46 chromosomes each. Okay, what's the process that's happening in the picture below? And we talked about this as a bell ringer. You have the first one, where is meiosis, and then it is fertilization, and then you have a zygote that has 46 chromosomes. Now, number 19 is kind of confusing. It says, which is the only possibility for the cell? Now, what would be a good key here if they had have said it is meiosis? But they did not. But you have to assume that it is. So what we're going to do is we're going to assume that it's meiosis. And then in that case, we're going to say that it starts with 4 chromosomes and it knocks down to 2. And because it has those number of chromosomes, one of each, so it has to be, without a doubt, it cannot be anything but A. Now, if it didn't tell us mitosis or meiosis, which it didn't, it could be that it could have been B because that is exactly the same amount, so it could be mitosis there. But indeed, the answer is A, as meiosis. Where would glycolysis occur in the cells? So here it is just asking you where at in the cells that glycolysis would occur, and the answer is in the mitochondria. Well, now you have to decide, is it in the matrix or is it in the inner membrane? Well, because glycolysis is the first step, we're just into the matrix, into the mitochondria, so this would just be in the inner membrane. The matrix would be later on into the electron transport chain. 21. The majority of the ATP produced during photosynthesis is produced during, and we've talked about this before in these recordings, it has to be glycolysis produces 2, Krebs cycle produces 2, and the electron transport chain produces a whole lot more. So the answer is C, electron transport chain. Okay, 22, put the following in the correct order. So we have to go and we look at these cells and we're going to put them in order. So we've got the interphase, and then we've got prophase, and then that's more like prophase, then no. Here we go. 2 is interphase, 3 is prophase, and then we have metaphase because they've lined in the middle. And then we go over to where they separate from each other, and this is anaphase. And then where they start to divide, that's telophase, where they start to form that cleavage furrow. And here is cytokinesis and telophase, which they've separated cytokinesis and telophase for you. But the main thing is from anaphase to telophase, these cells start to split, and you end up with 2 in mitosis. Okay, so find the mutation point on 23. Now, the thing here is all you're looking for is where there is a mutation forming. Okay, so we got GG, AA, TT, AA, CC, TT, CC. Here it is, G to A. Now, everywhere else it's been identical, but in this case, in section 3, there's our mutation point, and that's the mutation. Okay, so that would be C. Okay, which of the following is an example of DNA replication? DNA replication is where you are adding on A's and T's and G's and C's. There should be no U's, nothing like that going on. In this case, it would be A on my screen, but on your computer screen on Canvas, it shows C because somehow these letters messed up. So this is actually C, but this is the same picture. Okay. 25. Looking at the picture from the previous question, which is an example of transcription? Now, transcription is going from DNA to RNA, which means that you get rid of those T's and you should have U's. Okay, so in this case, the only other option we have is D. And I believe that's the same on yours. Yes, D. Okay, looking at the 26. What's the complementary strand to TACC? So that's simply what we've been doing all week. T to A to T, C to G, G to C, A to T. Okay, so in that case, 26 has to be A, and there's no other option to it. Okay, 27 is one of those that we haven't done. It's a Punnett square. So if you run a heterozygous, hetero means different. So if I run a heterozygous red and then a homozygous red, what's the possibility that the offspring of the plant will be a homozygous dominant, which means it has to have 2 capital letters with it. So in this case, 27 is going to be C. It's 50% going to be homozygous. And then over here, these are different. You have a capital and a lowercase, and in that case, it's going to be lowercase, uppercase and a lowercase, so that is heterozygous, and these are homozygous. So in that case, it'll be 50% heterozygous, 50% homozygous.